Hello friends and welcome. Today I will be acquainting you with starch and cellulose, which are plant polysaccharides. Carbohydrates occur in plant and animal tissues as well as in microorganisms in many different forms and levels. In plants, a wide variety of monosaccharides and oligosaccharides occur, and the storage polysaccharide is starch. Cellulose is one of many polymers found in nature. Wood, paper and cotton all contain cellulose. In the following sections, we will talk about starch and cellulose in detail. First, let us know about the occurrence and isolation of starch. Starch is widely distributed in various plant organs as a storage carbohydrate. As an ingredient of many foods, it is also the most important carbohydrate source in human nutrition. In addition, starch and its derivatives are important industrially. For example, in the paper and textile industries, starch is isolated mainly from the source such as corn, potato, rice, wheat, sweet potato, lotus root, etc. Recently, starches obtained from legumes, peas, lentils have become more interesting because they have properties which appear to make them a suitable substitute for chemically modified starches in a series of products. For the isolation of starch, the plant material is disintegrated. The starch granules are washed out with water and then sedimented from the starch milk suspension and dried. In other cases, such as cereals, the starch is embedded in the endosperm protein matrix. Hence, granule isolation is a more demanding process. Thus, a countercurrent process with water at 50 degrees Celsius for 36 to 48 hours is required to soften corn. That is steeping step of processing. The steeping water contains 0.2% sulfur dioxide in order to loosen the protein matrix and thereby to accelerate the granule release and the increase starch yield. The corn grain is then disintegrated. The germ, due to its high oil content, has a low density and is readily separated by flotation. It is the source for corn oil isolation. The protein and starch are then separated in hydrocyclones. The recovered starch is washed and dried. In the case of wheat flour, a dough is made first from which a raw starch suspension is washed out. After separation of fiber particles from this suspension, the starch is fractionated by centrifugation. In addition to the relatively pure primary starch, a finer grained secondary starch is obtained which contains pentosands. The starch is then dried and further classified. Now let us discuss structure and properties of starch. Starch comprises of two basic molecular components, that is amylose and amylopectin. These are identical in their constituent basic units, that is glucose, but differ in their structural organization, that is linkages. These variations in the linkages in turn affect their functionality in food applications. Amylose is a straight chain molecule, while amylopectin is a branched molecule. In addition, each is hydrolyzed, digested and absorbed differently. Amylose is hydrolyzed mainly by amylases, while amylopectin requires debranching enzymes such as pullulanase for complete hydrolysis. As a result of their structure and nature, and products of hydrolysis of amylose and amylopectin differ. The proportions of amylose and amylopectin in foods therefore influence the extent of digestibility of the starch. Amylose is a chain polymer of D-glucopyranosyl residues linked by 1,4 linkage. The molecular size of amylose is variable. Polymerization degree in wheat starch lies between 500 and 6000, while in potatoes it can rise up to 4500. This corresponds to a molecular weight of 150 to 750 kilodaltons. Amylose is the key component involved in water absorption, swelling, gelation of starch in food processing. High amylose starches are therefore most commonly applied in food products that require quick setting gels, such as candies and confectionery. Amylopectin is a branched glycan with side chains attached in the sixth position of the glucose residues of the principal chain. An average of 20 to 60 glucose residues is present in short chain branches, and each of these branch chains is joined by linkage of carbon 1 to carbon 6 of the next chain. The main proportion of starch granules crystalline structure is apparently derived from amylopectin. The organization of amylopectin molecules in starch granules is radial, the reducing end being directed outwards. Amylopectin, when heated in water, forms a transparent, highly viscous solution, which is ropey, sticky, and coherent. Unlike with amylose, there is no tendency towards retrogradation. There are no staling or aging phenomena and no gelling except at very high concentrations. Now, uses of starch. Starch is an important thickening and binding agent and is used extensively in the production of puddings, soups, sauces, salad dressings, diet food preparations for infants, pastry fillings, mayonnaise, etc. Cornstarch is the main food starch and an important raw material for the isolation of glucose. A layer of amylose can be used as a protecting cover for dehydrated and candied fruits, preventing their sticking together. Amylose treatment of French fries decreases their susceptibility to oxidation. Amylose films can be used for food packaging, as edible wrapping or tubing, as exemplified by a variety of instant coffee or tea products. Amylopectin utilization is also diversified. It is used to a large extent as a thickener or stabilizer and as an adhesive or binding agent. Now let's discuss different types of starch. Firstly, we will discuss a resistant starch. Starch and its degradation products which are not absorbed in the small intestine are called resistant starch. A resistant starch can, however, be metabolized by the bacteria of the colon. Acetic acid, propionic acid, and butyric acid are formed, stimulating the growth of cells of the intestinal epithelium. A distinction is made between 4 forms of resistant starches. Type 1, starch enclosed in cells, for example, coarse ground grain or legumes. Type 2, native starch granules, for example, in bananas, potatoes. Type 3, starch fractions produced on retrogradation, for example, in boiled potatoes, breadcrumb. And type 4, starch modified by Maillard reaction or caramelization. That is formation of glycosidic bonds which are not hydrolyzed by alpha amylase. Second type is modified starches. The properties of starch and its components can be improved or tailored by physical and chemical methods to fit or adjust the properties to a particular application or food product. Such starches are known as modified starches and include: number 1, mechanically damaged starches. These are obtained when starch granules are damaged by grinding or by application of pressure at various water contents. The amorphous portion is increased, resulting in improved dispersibility and swellability in cold water. Number 2 is texturants. Heating of starch to 100 to 200 degrees Celsius with small amounts of acidic or basic catalysts causes more or less extensive degradation. White and yellow powders are obtained which deliver clear or turbid, highly sticky solutions of varying viscosity. These products are used as adhesives in sweets and as fat substitutes. Number 3 is pre-gelatinized starch. Heating of starch suspensions followed by drying provides products that are swallowable in cold water and form pastes or gels on heating. These products are used in instant foods, for example puddings, and as baking aids. Starches undergo gelatinization and retrogradation, which is of enormous importance in foods. Let's understand what these phenomena are. When suspended in cold water, air-dried starch granules swallow with an increase in diameter of 30 to 40%. The process being referred to as gelatinization. If this suspension is heated, irreversible changes occur, starting at a certain temperature which is characteristic of each type of starch, called the gelatinization temperature. The starch granules absorb 20 to 40 gram of water per gram of starch, the viscosity of the suspension rising steeply. At the same time, a part of the amylose diffuses out of the granules and goes into the solution. Finally, the granule bursts In gelatinization, water first diffuses into the granule, crystalline regions then melt with the help of hydration, and finally swelling gives rise to a solution through further diffusion of water. In this process, hydrogen bridges between glucose chains in the crystallites are primarily separated, and perhaps some of those in the amorphous regions as well. Cooling of gelatinized starch results in the reassociation of the leached amylose from gelatinized granules. This is the process of retrogradation. This term denotes the largely irreversible transition from the solubilized or highly swollen state to an insoluble, shrunken, microcrystalline state. This state can also be directly achieved by slowly cooling a starch paste. The tendency towards retrogradation is enhanced at low temperatures, especially near 0 degrees Celsius, neutral pH values, high concentration, and by the absence of surface active agents. Retrogradation is also referred as setback and occurs with recrystallization of amylose. Amylose is much more susceptible to retrogradation, and amylopectin is only minimally involved in starch retrogradation, even though amylopectin has been shown to influence retrogradation and syneresis in cornstarch gels. This reassociation and recrystallization of amylose causes release of the water absorbed and bound during gelatinization, leading to the phenomenon known as syneresis. Retrogradation of starch in food products is a concern as it affects product quality. The stability of starch contained in products during cold storage, in particular, is greatly affected by the extent of retrogradation. Freeze-thaw crystals result in extensive retrogradation and syneresis. Retrogradation of starch in some instances enhances quality as such starches are resistant to enzyme hydrolysis. Now let's learn about cellulose. First, its occurrence and isolation. Cellulose is the main constituent of plant cell walls, where it usually occurs together with hemicelluloses, pectin and lignin. Since cellulase enzymes are absent in the human digestive tract, cellulose together with some other inert polysaccharides constitute the indigestible carbohydrate of plant food, that is vegetables, fruits or cereals, referred to as dietary fiber. Now, structure and properties of cellulose. Cellulose consists of beta-glucopyranosyl residues joined by 1,4 linkages. It crystallizes as monoclinic rod-like crystals. The chains are oriented parallel to the fiber direction and form the long B axis of the unit cell. The crystalline sections of cellulose comprise an average of 60% of native cellulose. These sections are interrupted by amorphous gel regions, which can become crystalline when moisture is removed. Cellulose has a variable degree of polymerization depending on its origin. It can range from 1000 to 14,000 with corresponding molecular weights of 162 to 2268 kilodaltons. Because of its high molecular weight and crystalline structure, cellulose is insoluble in water. In addition, its swelling power or ability to absorb water, which depends partly on the cellulose source, is poor or negligible. Now, uses of cellulose. Microcrystalline cellulose is used in low-calorie food products and in salad dressings, desserts, and ice creams. Its hydration capacity and dispersibility are substantially enhanced by adding it in combination with small amounts of carboxymethyl cellulose. Let's now have a glimpse of its derivatives. Cellulose can be alkylated into a number of derivatives with good swelling properties and improved solubility. Such derivatives have a wide field of application. Some of the derivatives are alkyl and hydroxyalkyl cellulose. The reaction of cellulose with methyl chloride or propylene oxide in the presence of a strong alkali introduces methyl or hydroxypropyl groups into cellulose, leading to its alkylation. The degree of substitution is dependent on reaction conditions. Mixed substitute products are also produced, for example, methyl hydroxypropyl cellulose or methyl ethyl cellulose. The substituents interfere with the normal crystalline packing of the cellulose chains, thus facilitating chain solvation. Hydroxyalkyl substituents stabilize the hydration layer around the macromolecule and thereby increase the gelling temperature. Changing the proportion of methyl to hydroxypropyl substituents can vary the gelling temperature within a wide range. Cellulose derivatives find diversified applications in bakery products obtained from gluten-poor or gluten-free flours, such as those of rice, corn or rye. The presence of methyl and methyl hydroxypropyl celluloses decreases the crumbliness and friability of the product, enables a larger volume of water to be worked into the dough, and thus improves the extent of starch swelling during oven baking. Since differently substituted celluloses offer a large choice of gelling temperatures, each application can be met by using the most stable derivative. Their addition to dehydrated fruits and vegetables improves rehydration characteristics and texture upon reconstitution. Hydroxypropyl cellulose is a powerful emulsion stabilizer, while methyl ethyl cellulose has the property of a whipping cream. It can be whipped into a stable foam consistency. Another one is carboxymethyl cellulose. Carboxymethyl cellulose is obtained by treating alkaline cellulose with chloroacetic acid. Properties of the product depend on the degree of substitution and of polymerization. Low substitution types are insoluble in water but soluble in alkali, whereas higher degree of substitution types are water soluble. Solubility and viscosity are dependent on pH. Carboxymethyl cellulose is an inert binding and thickening agent used to adjust or improve the texture of many food products such as jellies, paste fillings, spreadable processed cheeses, salad dressings, and cake fillings and icings. It retards ice crystal formation in ice cream, stabilizing the smooth and soft texture. It retards undesired saccharose crystallization in candy manufacturing and inhibits starch retrogradation or the undesired staling in baked goods. This was all about today's lecture. Thank you very much and have a nice day.